being joined now by Christina Toomey, who's a postdoctoral research associate at University College London. And we were just talking about brain banks.
Yes, I was listening, and I could add some more on that if you need to.
Yeah, for your work there. And Guliz, am I pronouncing this wrongly? Guliz Ozcan?
It's Guliz Ozcan.
Guliz Ozcan. Yeah. Oh, God, you know what? I'm useless with it. Anybody who's listening to the podcast will know that. Who's a postdoctoral research associate at also at UCL. Do you know Christina?
I do.
Yeah, we know each other. Yeah, we're both on the Alzheimer's Research UK UCL network. So we work together with that.
And there are so many people from UCL joining today. They've been amazing, my colleagues from UCL. And we've got Dr. Gemma Lace, who's a senior lecturer and lead for public engagement and outreach at University of Salford, representing the North.
Yes, Northern.
Well,
nice
I don't know if you saw earlier, we've had 3 researchers who were all talking about community. They were all from the North as well.
Excellent stuff.
Not that we're introducing a North-South divide in the life.
Absolutely not. We would not be encouraging that at all, would we?
Just me. I'm going to go round the table and ask you all to introduce yourself. So, Gemma, as we talk to you, could you go first?
Yeah, no problem. So, yeah, my name's Gemma Lace. I'm from the University of Salford. I've got a dementia research group, and I'm also head of public engagement and outreach. So, as well as doing the research thing, I like to go out and speak to the public and help bust some of the myths associated with dementia as well.
Fantastic, thank you. And Christina?
So as you said, I'm a postdoctoral research associate, and I'm actually based at Queen Square Brain Bank at UCL. So it's a fully fledged brain bank, and we use the brain donations to look at the different types of pathology that are occurring in the neurodegenerative diseases. And I also look at the proteomic profile, so the proteins present in these brains and the different levels and how that relates to pathology.
So you don't just collect samples to give to researchers, you perform research in your own right as the brain bank?
Yeah, so we have 2 aspects of the brain bank. So we have a diagnostic side, and anybody around the world can also request tissue from our brain bank. So it's not just us doing research on the brains that are donated to us, but we also have a team of researchers that are doing research themselves on the brain tissue as well.
Fantastic. And Guliz?
So I'm quite different from the previous panelists, I think, in the sense that we do basic research and we work with zebrafish. So I'm a postdoctoral researcher in the real lab, and the focus of our lab is sleep. So we use zebrafish, which is a small fish, as a model organism to understand the different genes or neurons involved in regulating sleep. And my particular project is about understanding how amyloid beta, the protein that makes the plaques in Alzheimer's disease brains, might be linked to sleep.
Fascinating. And they are, so I have to apologize because I did, I when I looked through the list of people that had offered to speak and tried to group people up, there were certain people that just were quite hard. And I have to say, you were the only person who came forward who said they worked on zebrafish, which made you quite difficult. So, but also super fascinating. Can I come to you first to talk to you about work? Explain to us as well, because I know we've got about 90 people watching us between Zoom and YouTube right now, and a few on the Periscope who might not quite understand the relevance of zebrafish to Alzheimer's.
So sure. So
talk us through it.
So from human studies, we can learn a lot, obviously, and they're directly relevant to the human condition. But we cannot always find we cannot always do mechanistic or causative studies. So it's important to have model organisms to go back to the genes and neurons that may be helpful to uncover mechanisms. And actually, I have prepared some slides that I can share. I will just give a little bit of background on Alzheimer's disease. Because there are different causes of dementia, and Alzheimer's disease is the most common cause of dementia. And as we know, it has symptoms that includes memory loss, problems with thinking or language. It's a physical disease that affects the brain, and it's defined by this extracellular amyloid beta plaques and intracellular tau tangles. So amyloid beta is basically a small peptide that builds up. And importantly, we can think of Alzheimer's disease in 3 phases. The first phase is the preclinical phase, where amyloid beta, the small protein that is thought to be one of the major drivers of the disease, starts to accumulate, but patients at this stage have no symptoms. And the second phase is when tau protein starts to aggregate and cell death starts to occur. This is known as mild cognitive impairment. And the third phase is Alzheimer's disease, where neurodegeneration starts to occur. And at this stage, the pathways and neurons might have been destroyed, and maybe this may not be very reversible. So it's important to kind of focus on these early stages, which may provide us with a window for early intervention, which may be successful before any irreversible damage is done. So I started to work on this project based on a few intriguing observations. One of them was about half of Alzheimer's disease patients had sleep-wake problems, and this was one of the early problems. So the number changes based on the study from 25 to 60, or 65. So let's say half of them have sleep problems, and this can be excessive daytime sleepiness, sleep fragmentation, and reduced sleep at night. So another intriguing observation was that physiological changes such as the sleep-wake cycle can alter amyloid beta levels in the brain. So this is actually human data, and I'm showing basically that the more time you spend awake, the more this amyloid beta starts to accumulate, and after sleep, its levels dissipate. And another observation was that sleep-wake regulatory neurons are lost in Alzheimer's disease. So people have come up with this hypothesis that if you have reduced sleep, you have more amyloid beta, and if you have more amyloid beta, your sleep is destroyed more. So there was this vicious cycle. So, but one of the missing pieces here was that sleep problems were actually one of the earliest symptoms, probably way before any major neuronal loss was happening. So we wanted to test whether amyloid beta's effect on sleep could be an acute function, and if so, what is the mechanism? So I'm not going to go into a lot of detail, but we can do these kind of more causal studies in model organisms such as zebrafish, and zebrafish has the advantage of giving us hundreds of eggs, so we can look at 5 to 7 day old zebrafish. We can track their behavior we can put them in these, in these 96 well plates and track them by a video camera and look how their sleep and wake patterns change. And because they're at the stage, their brains are transparent, we can also directly observe how their brain activity changes. So basically, I've done this work, and what we found is actually this vicious cycle was not as simple as originally proposed. We found that depending on the structure of amyloid beta, if it makes these small structures, it could reduce sleep. But if it made the longer forms, it could induce sleep via another protein that is actually involved in other neurodegenerative diseases called the prion protein. So I'm just going to summarize. So we found that amyloid beta can either increase or decrease sleep. And this is important because this suggests that accumulation of amyloid beta may be the underlying cause of the sleep problems in patients. So actually the pathophysiological pathways may be linked with the sleep pathways directly. So because this is sleep problems are an early symptom, this can give us a handle, like a time frame where we can intervene and be successful at that. So, and again, preclinical basic research can help us uncover such mechanisms, identify new therapeutic targets, or link, or help us uncover the mechanisms that lie behind phenomena that we observe. So that's all, and I'd like to thank AR UK because this study was actually funded by AR UK for 3 years. So we're really thankful for them to helping us to do this. And yeah, that's what I'm going to say. Thank you.
Brilliant. Thank you very much. I mean, I think you very eloquently there shared why zebrafish are important, which I don't think people necessarily would. I think it was quite a jump from there to people, but if it identifies those targets, and sleep has come up so often in the last 12 months about too much sleep is bad, not enough sleep is bad. You've got to, you know, it's a bit like the Goldilocks thing. You've got to have the perfect amount of sleep to make that you offset that with other risk factors. Gemma, let's come to you next. Can you tell us about your work? Is sleep important? Does that come into your research?
Yeah, specifically sleep doesn't relate to my research, but a lot of the research I do is associated with understanding risk factors. And when we think of dementia and the variety in dementia that we see when we look, for example, at people's brains, what I'm really interested in is firstly, why do brain cells die in people with dementia? And how can we maybe interfere with that process? What we know is that you get the buildup of junk in the brains of people with dementia, different kinds of junk associated with different types of dementia. And that doesn't happen in someone without dementia. So I'd like to think that me and some of you guys don't have a load of abnormal proteins clogging up our brain cells and causing them to die. And what my research is focusing on is why that happens and how that varies in different types of dementia. And what we found so far that is essentially, if you look at the different diseases that cause dementia, it's different waste disposal systems that are affected and give rise to this buildup of junk. You know, if you think about your own kitchen, you might get the buildup of waste because no one bothers to put the rubbish in the bin. It might be that you get the buildup of waste because people start putting stuff in the wrong bin. I've got 3 bins in my kitchen and 5 bins outside my house. Sometimes the bin doesn't get put in the right place. Sometimes the bins overflow because the bin men don't turn up. And so what our research has found is that within different types of dementia, it's a different issue. And that's really important therapeutically, because it means that we need to be thinking about personalised medicine approaches rather than looking for a magic pill that's going to cure dementia. We need to really be thinking on a personal basis about different risk factors. So things like sleep, things like obesity, smoking, how all these things come together upon with different genetic backgrounds to kind of create this recipe for dementia, and how we need to think about it on a individual basis.
And that's because there is general agreement now that there is no one single thing that is going to be the silver bullet or that causes Alzheimer's. It is going to be a collection of things, is what the evidence is showing so far. Yeah,
absolutely. I mean, we know that there are about 12 risk factors that we can actually target as individuals to lower our risk, and only about 1/3 of people understand that you can lower your risk of dementia by targeting things such as midlife obesity and, you know, hearing loss, diabetes, hypertension. But I'm making you feel bad now. Have you just eaten 27 bars of chocolate when you were behind them? It's all about balance, Adam. It's all about balance. But yeah, people don't understand that actually you can reduce your own dementia risk, and it is thinking about this kind of recipe. You've got your genetics, which influence, you've got the environmental factors, you've got your own lifestyle choices that you make, and all these kind of come together to impact your dementia risk. So some of them we can affect, some of them we can control, some of them we can't.
That's good. And then, so having identified the risk, I suppose it's then changing society to act upon that information.
Yeah, exactly. Yeah, and I think one of the things with dementia is for a long time people were under the assumption it was just an inevitable part of aging. It's just something that happened when you got older. But actually we know now that dementia is caused by a number of different brain diseases, and just like any other disease like cancer or
we can treat it if we understand more about it. And that's why Alzheimer's Research UK is so important. It has funded my research, it's funded most of the research of the people that are involved in today's session, and we need that funding to keep research moving forward.
Absolutely. Thank you, Gemma, and thank you to everybody that's donated so far today. Christina, let's come to you now. So you work in the brain bank, so that's got to be a fascinating place to be. Why don't you tell us about your work?
Yeah, no, it's definitely a fascinating place to be. People are always wowed when I tell them that I work with human brains, but it's such a valuable tool that people do give their brains to us, because as the other researchers have kind of alluded to, there's so much we still don't know yet about Alzheimer's disease and what happens, or any other types of dementia. So we can use this brain tissue to really look at what's happened in the brains of people that have had it. And at present, going back to the last session where they were talking about the misdiagnosis between people, because there's so much clinical overlap between symptoms of different types of dementia. We can only at the moment look at the pathology under a microscope to determine exactly what disease the person had in life, definitively. So part of my research is to look at the proteomic profiles of these brains and determine whether there's any signals, like protein-wise, that we can use to see in life what would lead to a certain pathology later in life. So without brain material, we wouldn't be able to do a lot of that research at all. So it's definitely a really valuable tool.
And do you use brain material, Gemma?
Yeah, I do. Yeah, that's why I'm nodding enthusiastically. I use brain. I've used human brain tissue my entire scientific career, and it is so useful for capturing that complexity of what is going on in a given individual's brain at any one time. I use cell-based models as well for my research, which is really good for trying to address very specific questions. But when you think about an individual and their entire life, everything that they've been exposed to, maybe the drugs they've taken, their lifestyle habits, their other comorbid diseases that they might have, as well as having dementia, you can get an idea of that from human brain tissue. And the Brains for Dementia research program it just helps out so many researchers with respect to making progress and looking at things from that bigger picture.
Because that's the valuable part of this, right? Somebody doesn't just get given a piece of brain tissue and then there you go, figure it out. That every sample comes with history. I mean, you come with potentially decades worth of history to go with it to understand. Do you capture things like what people were eating, what medications they're taking, other conditions that affected their life, lifestyle, exercise? I mean, how much do you capture? What goes with that piece of tissue?
Yeah, we get a really detailed clinical history from all of the donors that donate to us, and a lot of them have been treated at UCL, National Institute for Neurology, and they have a real big workup done on them, so that basically we have all the information that we can possibly have about these brains that we do get given to us. And basically one of the projects that I'm working on now is looking at pathological aging. So there are some cases where people will have some of this buildup of Alzheimer's type pathology in their brains, but they never get any symptoms during life. So they're actually termed as healthy when they die. But it's really interesting why they do still have some of this pathology in their brains. And we're looking back at all the records, all of them past medical records, and seeing if we can make a correlation between something that these patients have had that has led to them still being able to live a healthy life,
complete sense,
versus those that do go on to get dementia symptoms.
And that's why you collect brains from people that don't, not just people with dementia, but without as well.
Yeah,
so
very important.
You can collect all that and then give that to Gemma, who as well as doing your own research as well, and you can look at, start to unpick this. We've got some questions, and we've only got about 4 minutes left, so I'm going to whiz through these questions. Okay. Gemma asked this actual question was put to the previous researchers, but I'm going to ask it to you because I think it's relevant. Have you found difficulties getting funding because of COVID? Have you?
Yes.
So fine if you're a COVID researcher, right? If you could suddenly turn your hand to COVID research, there's been lots of research for COVID, but not for the stuff you were doing anyway.
Absolutely. It's important to acknowledge that COVID has had a huge impact on families with dementia, living with dementia. For someone not living with dementia, it's been quite an isolating experience at times. And so for families impacted by dementia, it has been difficult. And so we still need to be pushing to fund to keep things moving forward. An example is I got told that one of my projects was in the fundable range, and it should have really been getting the money through, getting the staff through. Now September, October time, I'm still waiting now because everything's had to pause. Everyone's had to take a breath. Everyone's had to have a revisit of the funding.
Yeah.
And it's a shame because I know a number of researchers who are desperate to carry on their work, but the funding is disappearing. So yeah, we've definitely been affected.
I was about to apply for fellowships. So for those that are listening, when you start to gain your own independence as a researcher, you need to apply for fellowships to get your own funding. And it's an important part of building your own career. And a lot of so Alzheimer's Research UK, Alzheimer's Society, all of the typical dementia research funders have had to pull their funding for that type of grant because there just isn't the funds at the moment due to the current pandemic, which is really unfortunate. But hopefully in the future, through events like this and things like that, we can get more funding again.
And the Brain Bank, of course, I'm assuming the Brain Bank's been able to still collect its donations or not?
So they weren't for a while. They did have to stop as well because the mortuaries couldn't actually process any of the brains because they were dealing with obviously a lot of COVID patients. But they are back up and running now. We are collecting again, and we're back fully functional. So it is getting there again.
Yes,
we've got a different question, Julius. We've got a different question for you because we've only got 1 minute left, and you've had a specific question asked. So I'm going to take it. It's tricky for you too. I think. Is that fair? Just nod. Yeah. Everybody's okay. Inma asks, "Hi, a question for Julius. Is there any biomarker in blood, CSF, brain in people that do not sleep?" Difficult question, I know, brackets. You're on mute.
So I think there are some, I think they can detect, for example, orexin peptide, but I think you can have, so the problem is, again, with sleep, we don't know all the pathways or proteins that are involved with it. So, so I'm not 100% sure whether there is one biomarker that can detect all the sleep problems. I don't think there is.
No. So last point. So Lucy Sharple says, "Hi Julius, thank you very much. Good talk." Thank you very much. Rebecca Daly, who's going to be talking later on from Dementia UK. It's a great way of explaining it, Gemma. I completely agree as well. I thought you all did fantastically well at explaining because you all are highly technical, highly professional people at working complex things, and I think you did an amazing job at explaining your work really succinctly in a way that I understood it, so I'm sure everybody else did too. Thank you very much. And just to make a point, so you are all now back to work and continuing and as best as you can.
Yep, I'm in the lab right now.
Well, Well, thank you very much to all three of you for joining us over the last half hour. I'm really grateful for you taking time to share. As I've mentioned before, for everybody else, all the details of our speakers can be found on the website. There you'll find details of their Twitter. Go give them a follow. And if you have a question that you wanted to, you didn't get a chance to ask today, or if you're watching the recording of this later on, after the 7th of September, please do reach out, make contact through Twitter, and I know they're all very approachable and very helpful. And if you're interested in finding out more about Brains for Dementia research particularly, and you'd like to donate, they've got a website, and I know that they have, we should add, I know you mentioned UCL, but Gemma, the brain bank is there's a brain bank in...
Yeah, we use the Manchester Brain Bank, and
brain
also
banks,
we work with Sheffield Brain Bank as well. So
there are brain banks all over the country, overseas as well, if you're not watching in the UK, and certainly in Scotland as well. They all have different criteria for who can donate, but I think it's very valuable. So please do take a look at that. Thank you very much, everybody.